Good morning, everyone. Thank you for being here on such a nasty morning. Before we continue our discussion of nucleotides metabolism, I would like to make an announcement. I would like to invite all of you to come to tomorrow's Chem Club Chem Demo Night. It will be at 6:30 in Faraday Hall 143, so that's the big lecture hall in the old part of the chemistry building. I would also like to encourage you to consider joining Chem Club if you're not ready at the end of the semester. We desperately need of new members. Lots of our active members are actually graduating at the end of the semester, and we hope to be able to continue doing all the outreach activities and fun activities to engage the community in chemistry actually in the future. So we have a general meeting last Wednesday of this month, will be the last meeting before the fall semester, and you can learn more about Chem Club by just asking me. I'm the faculty advisor of Chemistry Club, or any of the Chem Club members are just coming to our next general meeting. And we have free pizza and soft drinks. It's a lot of fun. Okay. So, we talked about the so-called de novo synthesis of purine nucleotides last time, and I just wanted to give you an idea how complex this synthesis is. And I told you that I at least expect that you know where these atoms that make the purine ring system come from, which was elucidated, as I told you, by John Buchanan using radioactively labeled precursor molecules that he fed to birds. So I also told you that this de novo synthesis of purine nucleotides is very cost intensive, depending on the organism that we're looking at and whether or not at one step ATP is actually required. We need something like 7 or 8 or 8 or 9 AMPs or ATP equivalents for the synthesis of AMP and GMP respectively. So this is very cost intensive, and for that reason it's also important that the purine ring system, if it is actually produced in degradative processes when DNA and RNA is degraded, is also reutilized. And this can be done in so-called salvage pathways for purines. And I think the term salvage pathway is not really describing what is happening here, because we're just talking about relatively simple and straightforward enzymatic reactions that link appropriate purine nitrogenous bases, namely adenine, hyperxanthine, or guanine, to a phosphorylated ribose. Okay, so we just form a nucleotide again from these nitrogenous bases using phosphoribosyl pyrophosphate. So this was the starting material that we also learned about when we talked about the de novo synthesis of purines. So there are two enzymes that can form the appropriate nucleotides. Adenine phosphoribosyl transferase can form AMP by just linking adenine to a phosphorylated ribose, utilizing PRPP, phosphoribosyl pyrophosphate, as a substrate. And then this enzyme here, hyperxanthine guanine phosphoribosyl transferase, can either utilize hyperxanthine. Remember, that was an intermediate in the synthesis of AMP and GMP, or guanine to form inosinate or GMP respectively. Now, these enzymes obviously are of great importance for physiological systems, okay, so for us humans, for example, but in particular this second enzyme, hyperxanthine guanine phosphoribosyl transferase, has also found very important applications in, let's say, biotechnology, because it is important in the production of monoclonal antibodies. So I'm not quite sure how much you know about antibodies. I may have mentioned them briefly in this class. We may have talked about them in other classes. So antibodies are proteins that are produced by the immune system of vertebrates, like humans, okay? And they bind to foreign materials that invade a body, let's say a protein or bacterium or something like this, that is recognized as being foreign, can be bound by antibodies, if they have the appropriate binding specificity, and are in this way marked for destruction by other components of the immune system. Okay? So that's all that antibodies really do. They just bind to foreign materials, and in this way let the immune system know that this is something that doesn't belong there, it's foreign, and should get removed one way or the other. Okay? So if, for example, you have foreign protein in your body, you have a cut and something enters your body, and let's say it's a protein for simplicity, then there are different sites on this protein structure that could be bound by antibodies having different specificity. Okay, so these different regions that could be recognized by different antibodies are called epitopes. And each antibody in your system, a population of antibody, has a particular binding specificity. And there are millions of different specificities that is displayed or are displayed by the different antibodies that you have in your system. So let's say we have in a protein 25 or 100 different specificities, different epitopes. They could be bound by different antibodies having different specificity. Then these would be amplified. You could have so-called plasma cells producing these antibodies, and then these antibodies that would be in your bloodstream or other tissues or bodily fluids would bind to this foreign material and mark it for destruction. Now in biotechnology, let me take a step back. So since your body, your blood, then contains potentially a multitude of different antibodies directed against this target, this protein, your blood actually is polyspecific for this protein. So you have different antibodies, polyclonal antibodies, because they were produced by different plasma cells, each of which produces an antibody with one specificity, that all could bind to, let's say, the 25 different epitopes on this protein. Now, in biotechnology, it may be important to have just one antibody being produced in cell cultures that produces, or that has one particular specificity that is desired. And these antibodies then, that are produced in cultures, in cell cultures, are actually monoclonal antibodies. Monoclonal because they are produced by one type of cell. Okay, one clone of cell. And Köhler and Milstein actually demonstrated for the first time that you can produce such monoclonal antibodies by fusing antibody-producing cells, lymphocytes, plasma cells, with cancer cells, and make these clones of cell lines immortal. Okay, because the cancer cell would just continue to divide, and the plasma cell, a lymphocyte component of a fused cell, will then be able to produce an antibody having a certain specificity. Okay. And what you want to do is you want to fuse these cells. There are different ways how you can fuse them. This is just a picture, sorry it's a little blurry, that shows two cells that are about to fuse. Okay. And you can induce this either with certain viruses. That's what was used originally by Köhler and Milstein. They were awarded the Nobel Prize for this work. Or more easily with polyethylene glycol. So I have produced many monoclonal antibodies in my career, and what you use actually to then cultivate these cell cultures is a particular medium, fat medium, that contains amylopterin, hypersensitive amylopterin, and thymidine. And the reason why you do this is because you want to just select for cells that are fused and are fused of lymphocytes that produce an antibody with a certain specificity and cancer cells. And you don't want to cultivate, for example, just cells that are cancer cells or are fused cells that contain just cancer cells, and you also want to get rid of lymphocytes that are not fused with a hybridoma cell. Okay, so the lymphocytes, because they are mortal, will die off after a couple of days of cultivation anyway. The cancer cells, however, wouldn't die off unless you use cancer cells that are deficient in this enzyme, hypoxanthine guanine phosphoribosyltransferase transferase. And using this medium, which contains aminopterin, which is an antifolate, that knocks out dihydrofolate reductase, makes these cells that are deficient in this enzyme mortal in a medium that doesn't allow them to do a de novo synthesis of purines. Okay? Because you have aminopterin in the medium. This is an antifolate that knocks out the de novo pathway. So these cells cannot synthesize purine nucleotides from scratch. And then the cancer cells that you use are deficient in this enzyme that would allow these cells to produce inosinate or GMP in the salvage pathway. It means that these cells that have this deficiency in this enzyme cannot survive in such a medium. Okay? So if you do a fusion now between lymphocytes and these cancer cells, your lymphocytes that are not fused with a cancer cell will die off anyway after a couple of days. The cancer cells that are deficient in this enzyme, and that's a type of cells you use in these fusions, will die off because they can't synthesize in this medium, which contains aminopterin, purines from scratch, and they can't produce inosinate or GMP utilizing this salvage pathway because they're deficient in this enzyme. That means that they will also die off. And the only cells that should survive in this medium are those that are produced, those hybridoma cells, the term to use here, hybridoma cells, that contain of a lymphocyte, which still has this enzyme here, okay, and immortal cancer cells. And so if you cultivate your fused cells for a couple of days, then more and more cells will die off, and what should survive are those that are hybridoma cells resulted from the fusion of a lymphocyte and a cancer cell, and those you cultivate then and test for the production of antibodies that have a certain specificity, and you can use a great variety of different tests actually to do that. So this enzyme here actually is of great importance here for the production of monoclonal antibodies. Okay, so with this we come to the synthesis, de novo synthesis of pyrimidine nucleotides, and here you see a similar picture of what we saw for purine nucleotides that shows us where the individual atoms that make the pyrimidine ring system come from. And obviously we are talking about a smaller molecule, so there are fewer atoms actually that make this ring system, just a six-membered ring. And we see that actually 4 of these 6 atoms that make this ring come from one molecule, namely from aspartate. One comes from glutamine side chain nitrogen, glutamine amide group, and then amino group, and then one comes actually from bicarbonate / carbon dioxide. So also the synthesis of pyrimidine nucleotides is much more straightforward compared to those reactions that led to the synthesis of purine nucleotides. And it starts with a reaction that looks somewhat familiar, which produces carbamoyl phosphate. So we've talked about the synthesis of carbamoyl phosphate in different contexts. Do you remember what context?
Urea cycle.
Urea cycle. Okay, can you tell us a little bit about the synthesis of carbamoyl phosphate in the urea cycle?
It's the first substrate made in the urea cycle.
Mm-hmm. Do you remember where the nitrogen came from? What is the urea cycle about? Huh?
I think it's, wasn't it? It uses glutamine dehydrogenase to obtain free ammonia.
Exactly. So we produce in the liver ammonia, okay, and the ammonia is incorporated into the carbamoyl phosphate by which enzyme? Do you remember? Just look at the name of this enzyme, and then you get a clue. One. Carbamoyl phosphate synthetase 1. Okay, remember, whenever we encountered enzymes that had this 1 in their name, we concluded, rightly so, that there should be a number 2 also, and the same is true here for carbamoyl phosphate synthetases, because the enzyme that participated in the urea cycle, participates in the urea cycle is carbamoyl phosphate synthetase 1, which produces carbamoyl phosphate utilizing nitrogen from ammonia. And also, do you know the location where the urea cycle actually occurred?
Mitochondria.
Mitochondria, okay, so the mitochondria of the liver. Now here, actually, when we talk about the synthesis of carbamoyl phosphate as it relates to the synthesis of pyrimidine nucleotides, it's a little different because it occurs in the cytosol, okay, and also utilizes the nitrogen from a glutamine. Okay, so the nitrogen source is different here. It's not free ammonia, but it's a amino nitrogen from the side chain of glutamine. Okay, but the molecule that is synthesized just is the same as what we saw in the urea cycle as carbamoyl phosphate. So remember that in the urea cycle we actually then had an attack of this carbon here, activated with cleave of phosphate. Same happens here, aspartate actually, so it's aspartate here, okay. Not ornithine, but aspartate, the alpha amino nitrogen of aspartate actually attacks this carbon here and produces carbamoyl derivative of aspartate, utilizing trans-carb amylase, okay. And then we have an enzyme that hydroporotase that cyclizes this structure. Remember, we want to synthesize a pyrimidine nucleotide. Then this is being reduced to produce pyrimidine orotate. And if you look at the structure here, there are, let's say, two things that are noteworthy, I would say. First of all, this is an isolated free pyrimidine base that is being synthesized. Remember when we talked about the de novo synthesis of purine nucleotides? Free purine nucleotides aren't synthesized in the de novo pathway, but it's purine bases, excuse me. But it's a purine nucleotide that is being synthesized because we build up the ring system linked to phosphorylated ribose to begin with. Here it's different. We first synthesize a pyrimidine base, and if you look at the structure, Do you recognize this structure? Can you tell me what it's based on? If you just remove this carboxyl group, it's uracil. Okay, so this is carboxylated uracil that is produced here. Here we are. It's called orotate, and this orotate is linked to phosphorylated ribose utilizing phosphoribosyl pyrophosphate again. We cleave off inorganic phosphate, so this pyrophosphate. So this pyrophosphate again just served to activate the anomeric carbon to facilitate the transfer of this phosphorylated ribose to the nitrogenous base, or vice versa, to form this N-glycosidic bond between the nitrogen number 1 and our anomeric carbon and ribose. So at this point we have produced a nucleotide. Okay, we have a nitrogenous base, we have a ribose, a sugar, and we have a phosphate. But it's not one that you are probably overly familiar with. Okay, it's none of these pyrimidine bases or pyrimidine nucleotides that we usually find in DNA or RNA. But we can produce uridylate or uridine monophosphate, but just removing this carboxyl group here and converting this ring system into a uracil, and we have produced our first, let's say, common pyrimidine nucleotide, UMP. So this can be phosphorylated to produce a triphosphate utilizing appropriate kinases to produce UTP. And UTP, if you look at the structure, isn't too different from CTP or uracil from cytosine, because we just have to replace this oxo group here with an amine, and this can be done with so-called CTP synthetase, which utilizes energy in the form of ATP and again the side chain nitrogen from glutamine to incorporate the nitrogen here. Okay. So we have produced UTP and CTP at this point. These are both ribonucleotides. Okay, here we have this. Let's go to this first. We also need deoxyribonucleotides for the synthesis of DNA, deoxyribonucleic acids. So the ribonucleotides can readily be reduced to the corresponding deoxyribonucleotide with the help of an enzyme called ribonucleotide reductase. I'll come back to that in a moment. Let's first take a look at the regulation of the synthesis of pyrimidine nucleotides. So don't expect you to remember any details here. Same principle that we saw for the regulation of the de novo synthesis of pure nucleotides applies, namely that our products at one point or another actually have a negative feedback or exert a negative feedback regulation. So you see the CTP can actually inhibit here this step here where we produce our carbamoyl aspartate. That happens in E. coli, for example. We have UTP and CTP actually act on carbamoyl phosphate synthetase 2 in animals. We also have inhibition here of UMP onto this enzyme that removes the carboxyl group from our carboxylated uracil, so to say. And then we also have a couple of activating activities here. What is interesting, I think, is that ATP actually activates here this carbamoyl phosphate synthetase 2, and the rationale behind this is that if we have higher concentrations of ATP and in general purinucleotides, the cell realizes, okay, it's probably time to synthesize nucleic acids, and we also have to kickstart our synthesis of pyrimidine nucleotides. Okay, so let's come back to these deoxyribonucleotides. And, at this level, when we produce the deoxyribo compounds, we have actually diphosphates. Never mind, they can be converted easily to triphosphates with the help of kinases. So this, I want you to realize, is a reduction reaction. Okay, so we remove an oxygen here at the 2' carbon. Okay, so this is a redox reaction, and we always have coupled reductions and oxidations. So if this is a reduction, something else needs to be oxidized, and it's actually two sulfhydryl residues that we find in the enzyme, in the ribonucleotide reductase, which are then forming a disulfide bond. In order to regenerate the reduced form of these two cysteine residues, we can go through two possible pathways. We can either utilize so-called glutaredoxin or thioredoxin. So both of these are peptides / small proteins that contain also sulfhydryl residues, which can change between the reduced sulfhydryls and oxidized disulfide. So this can be utilized to regenerate here our reduced sulfhydryl groups in ribonucleotide reductase. And then there are certain differences how these functional groups are reduced. So in the case of glutaredoxin, there's a reductase that utilizes glutathione. We talked about the structure of glutathione just last week or so. Do you remember what glutathione looks like? What is it structurally? Any idea? Yep.
Isn't it like a gamma glutamyl cysteine glycine? It's like a tripeptide.
It's a tripeptide. Okay, if we want to be real correct, you would indicate that the carboxyl groups in the gamma glutamate and the cysteine are consumed in the formation of peptide bonds by indicating this by adding the ending -yl. Okay, so gamma glutamyl cysteine glycine, but otherwise you are completely correct. Okay. Yes, it's a tripeptide. Okay, remember that this side chain sulfhydryl group coming from cysteine can be oxidized by reacting with another glutathione. And this happens here, for example, in this reaction here that we see on this slide. And then, in order to reduce glutathione again, there's an enzyme, glutathione reductase, that utilizes NADPH to accomplish this. If we look at this side of the slide, the thioredoxin, the oxidized state, can be reduced back with the help of a reductase that utilizes FADH2, and this can actually be converted back again after oxidizing it to reduced form, also utilizing NADPH. Okay, so we're still talking about synthesis of pyrimidine nucleotides, and we have seen how we can form CDP, how we can form UMP, phosphorylating these would form CTP and UTP. Okay, that's all straightforward. Just talk about how ribonucleotide reductase can convert this into the corresponding deoxyribonucleotides that could be used for DNA synthesis. Okay. They can also be phosphorylated to convert them to the triphosphate. A deaminase actually can also convert CTP to DUTP. We can cleave off also phosphates. But what is noteworthy here on this slide is that DUMP can readily be utilized for the synthesis of the TMPs. Okay, so we haven't really talked about the synthesis of thymidylate / deoxythymidylate. And if you look at the structures of uracil and thymine, do you remember what the difference is? And I mentioned this in different contexts previously. Thymine is simply a methylated uracil at carbon number 5. Okay, so all we have to do is introduce the methyl group at carbon number 5 here in this DUMP, and that is done with the help of an enzyme called thymidylate synthase. And you see that, okay, this is our uracil ring, this is our thymine ring. The difference really is just this methyl group here. And what we obviously need is a cofactor that this enzyme can utilize that is capable of transferring 1 carbon units. Okay, so theoretically, nature could have chosen acetanosyl methionine for this job. Didn't. Okay, we have actually a derivative of tetrahydrofolate, namely the N5 and 10 methylene tetrahydrofolate, that provides this 1 carbon unit, also provides a hydrogen to actually make a methyl group, and then we convert this DUMP into DTMP. Okay, so this is also relatively straightforward. Remember that this N5 and 10 methylene tetrahydrofolate is the major entry point into our pool of tetrahydrofolate derivatives and can be produced in this reaction that we already discussed for the synthesis of glycine from serine. We talked about synthesis of non-essential amino acids, you remember that? And this reaction here, catalyzed by thymidylate synthase, as you see, that produces actually a partially oxidized tetrahydrofolate, a dihydrofolate again. Remember when I introduced tetrahydrofolate, I said it's derivative of folate, vitamin that we have to take up with our diet, okay, and is reduced by an enzyme called dihydrofolate reductase. First to dihydrofolate and then to folate, the active, to tetrahydrofolate, the active cofactor. So here, in order to produce the active form of our cofactor, we need this enzyme here again, dihydrofolate reductase, which utilizes NADPH, to fully reduce dihydrofolate to tetrahydrofolate. Okay, so here's another example where dihydrofolate reductase is important, namely in the synthesis of thymidylate. So if you mess with this enzyme, dihydrofolate reductase, inhibit it utilizing so-called antifolates like aminoterin and methotrexate, you not only knock out the activity of this enzyme, but you also mess with the synthesis of nucleotides and with this, with the synthesis of DNA. And if a cell wants to divide, okay, it can produce DNA, it won't divide, and ultimately these cell lines will die off. And this actually is the idea of using antifolates and inhibitors of this enzyme dihydrofolate reductase in chemotherapy. And I told you previously that antifolates are among the oldest chemotherapeutics.